In this concept, enzymes and metabolic reactions, we will be tying what we have learned about metabolic reactions to how enzymes help in all of this. The topics will be the activation energy barrier, how enzymes lower the activation energy or EA barrier, the active site of enzymes, substrate specificity, and the effect of local conditions on enzyme function. Even for a high energy covalent bond, it takes energy to break that bond. The energy required to convince two atoms to stop sharing the electrons in a covalent bond is called the activation energy, or EA. Once we get over the EA barrier, the reaction can proceed. Heat can provide the energy, causing the atoms to vibrate until they move far enough apart that they are too far apart to share electrons, thus breaking the bond. But if you are a cell, you want to be careful about building fires inside your house, so to speak. Cells don't have fireplaces. So instead they use special proteins called enzymes to lower the energy of activation. That way you can break molecules apart without setting the cell on fire. To explain how enzymes do this, I'm going to use a tool analogy. Suppose I needed to tighten a screw on my glasses back before I had LASIK performed. I could use my fingers, but I don't have strong fingernails and they really don't do a good job. The activation energy was too high for me to accomplish the job of tightening the screw in a timely and efficient manner. So what could I do? I got a screwdriver, one small enough to fit into the groove of the tiny screw. Then I could turn the screw easily. Enzymes work in a similar way. The reactants in the reaction, or substrates of the enzyme, fit neatly into the enzyme. Where the substrates or substrate of an enzyme fits is called the active site. The theory with the most supportive data states the binding of the substrate or substrates into the active site causes the enzyme to change shape. This puts pressure, so to speak, on the bonds of the substrate or substrates, lowering the activation energy and making the bond more easily to break. Once the bond is broken, they don't fit in the active site anymore. The enzyme spits them out, so to speak, and is ready for more substrate in its active site. This diagram shows how an enzyme could lower the activation energy in an exchange reaction. Let's go back to my glasses example. Could I effectively use a hammer to tighten my screw? Not if I wanted to be able to see out of my glasses afterwards. Just like tools for fixing glasses, enzymes are very specific for what they can be used for. They have substrate specificity. In other words, most enzymes only work on one substrate or substrates. If you look at this model of an enzyme binding its substrate, you can see how the active site fits the substrate exactly. The enzyme fits the substrate just like the tool fits the job. You can also see how the enzyme changes shape once the substrate is bound. Certain external conditions affect the functioning of enzymes. For example, as this chart shows, the human enzymes work best at about 37 degrees C. That is exactly the temperature of the human body. If I were to drop your core temperature, the enzymes don't work as fast and you slip into hypothermia. If I raise your core temperature up to about 42 degrees C, the enzymes in your brain start to denature and you go into convulsions and die. But not all enzymes function at the same temperature. The enzymes found in heat-loving, hot spring-dwelling bacteria operate best at the temperatures of the hot spring. Interesting. Each organism, humans and hot spring-dwelling bacteria, have evolved enzymes that work best at the temperature the cell finds itself in. Here's another example. You have in your stomach an enzyme that breaks down proteins. It is called pepsin. If you had to guess, what pH would it work best at? That's right, a pH of 2. Now the enzyme in your intestines work best at about a pH of 8, 7.5, somewhere in there. If pepsin moves from the stomach to the intestines, which it often does, it stops working. Trypsin, an intestinal enzyme for breaking down proteins, takes over. Well, that's it for this concept. Just one more concept to go.